everyone, welcome to the 13th video of 100-day Biopharmaceutics challenge, whereby we are going to cover the phases of biotransformation. Till now we have seen what exactly is the term biotransformation means. Now let's see what phases it has. So basically the point here is to convert the molecule into hydrophilic molecule so that it is easily excreted out of your body. That means there are phase 1 and phase 2 reactions which are going to help convert this molecule into the metabolite which is in the more excretable form, right? So for example, let's take an example of a drug or a metabolite that passes through liver. It passes through phase 1 reaction. Now in the phase 1 reaction, we also call them as functionalization reaction, right? Which means that we have added some functional group so that the metabolite becomes polar and is more available to be excreted. So phase 1 metabolite that is formed will be easily we can say in the hydrophilic nature and ionized now, right? Now this metabolite can be excreted directly or it is passed to some phase 2 reaction. In the phase 2 reaction we call them as conjugation reactions. So phase 2 reactions actually add a conjugating agent to the metabolite which is going to guide it and convert it into more hydrophilic, right? So the phase 2 metabolite is formed. This metabolite is formed by addition of a conjugating agent, right? So we ultimately have to see which ones are the phase 1 reactions, which ones are phase 2 reactions. So the ultimate metabolite that is formed is in the ionized fraction. That means the solubility got increased, the polarity got increased, which makes it more into the easily to be excreted. So in the phase 1 reaction, remember orchard, right? We have oxidation, reduction, hydrolysis and cyclization reaction. In the phase 2 reaction, remember games. We have glutathione conjugation, we have glucuronide conjugation, we have amino acid conjugation, we have acetylation, we have methylation and sulfonation. I'll write it here. We have glucuronidation. These are all based on what kind of your conjugating agent is going to bind. Glucuronic conjugation, it is glutathione conjugation. We have amino acid conjugation. We have acetylation. We have methylation and sulfonation. Alright, so all these reactions are actually in a manner that these are going to help bind an endogenous molecule, making them more polar, right? Let's see a little more about these phases. So in these phases, yes, these are phase 1, phase 2 reactions, but this is the major slide where we need to focus. As I mentioned, in the phase 2 reactions, we are going to let them bind to a conjugating agent. For example, you can see in the first reaction, which is the glucuronidation, we have a conjugating agent known as the glucuronic acid using the enzyme UDP glucuronyl transferase. This is the inactive intermediate. So it is ultimately going to bind to the glucuronic acid as conjugate. So you can see in this sulfation we have sulfate. In the amino acid conjugation we have glycine. In the glutathione we have glutathione as conjugating agent. In the acetylation, very important, we have acetyl coenzyme A. In the methylation we have L-methionine. So these conjugating agents are now I think clear to you all what are they going to do, but these are going to bind and the extent to which these reactions occur, let's see those. But for an exam point of view, this reaction, their transferring enzyme and their conjugating agents are very important. One more additional point I would like to mention, that the genetic polymorphism is associated with acetylation. This question has been asked multiple times in different ways. So acetylation is the reaction in which the enzyme we have N-acetyl transferase. This has a role in the genetic polymorphism. Please note it down as well. So if somebody asks you that what is the order of the capacities of these conjugation reactions, so number one is going to be the glucouronidation reaction. Then you have is the amino acid conjugation. Then you have is the sulfation. Then glutathione conjugation. That is why these phase 2 reactions are called as conjugating reactions, as phase 1 reaction are known as your functionalization reaction, right? So I have told you that in the glucouronidation we have glucouronic acid as the conjugating agent and the enzyme is UDP glucouronic transferase. In the sulfation we have sulfate as the conjugating agent. Transferring enzyme will be sulfotransferase. From the name itself it is clear. So we have different transferase enzyme for it. We have amino acid conjugation in which glycine will be the conjugating agent and transferring enzyme will be acyl transferase. Similarly, in the acetylation, it will be N-acetyl transferase. We have glutathione in which it is going to be the glutathione as the conjugating agent and enzyme will be glutathione S transferase. Next, we have acetylation. This acetylation agent is going to bind with the convert with the N-acetyl transferase. So here it is written that conjugates are excreted in the bile. Why? Because if you remember, if we talk about the excretion part, the drugs with a lesser molecular weight actually excretes through urine. But while they are binding with a conjugate, their sizes are going to increase so that they are excreted in more in the bile. So this is about the phases of conjugation and the your phase 1 and phase 2 reactions of the metabolism. I hope you had a good time recognizing these reactions, their order of capacities and all the enzymes and the conjugating agent in between. Thank you so much. We'll meet in the next session.